Morning, and thank you for introduction, and thank you for giving me the opportunity to present my work here. And I started work on the microbiome in lab. First time I stabilized all the work here. So this is the preliminary study on the rat, and in the lab we have a very stabilized model of the rat for the enrichment environment or non-enrichment environment. So this reflects the community to the low socioeconomic communities and the rich socioeconomic community. And thank you, Marielle. She made my presentation very easy because she introduced the what are the difference in the low socioeconomic and the higher value. And unlike the Alec, my work is completely on the to show the behavior data. And my next step is to go in the future to study the RNA-seq and the methylation level. So with that, so here, in this paper published in 2020, and they studied the cognitive test scores and the development of the brain in the population in the low income and the high income. And this data clearly showed that in the low income people, the cognitive test scores is lower compared to the high income. And for the brain development and the volume is directly correlate to the cognitive test scores. So the volume of the brain is lower in the low income and higher in the high income. So, so first, this experiment was done in 1972, and they showed that what is the difference for difference in the brain development in the impoverished, non-rich environment versus the enriched environment, and they divided the mice, and they put the one mice in the single cage to show the non-enriched environment, and multiple mice in the big cage, and with the toys and the other engagement activities to the mice. So it showed clearly that the brain branching of the dendritic cells in the brain is more in the enriched environment. So combined with these two, enriched environment published in the 2020, and this was the first experiment to show that there is a difference between the low enriched and enriched environment in the development of the brain. So we developed in the lab the exposure of the lead PB in the rat model in the early post natal from the post-natal day 0 to 21. We provide the lead diet to the mothers and throw their milk to the pups. So the exposure of the lead to the pups through the milk of mother. And we designed the to show the non-enriched and the enriched cage. We put the after weaning, put the pups in the cage, non-enriched and the enriched cage. So, but here in our lab to develop the model, we put the 3 rats in a smaller cage with no toys or activity to show the non-enriched environment. And for the enriched to engage the pups, we put the 6 pups with the toys for the engagement of to do the uh, to engage in the social activity. And then after 55 days, putting in the enriched environment and the non-enriched environment, we tested those rats for the behavior test. That's a trace fearing conditioning test. In this test, there is a sound, and after 20-second trace fearing time, there is a shock. And we this called the acquisition. So this is tone and the shock to acclimatize the pups 6 times, and then we put the pups to test their memory for 1 day, 2 day, and day 10. So 1 day is too early. They're supposed to memorize in 1 day all of them. Day 2 and day 10 is the real test. If they memorize the tone and the shock. So after that tone, if they remember correctly, after that tone, they should, there is anxiety. They memorize that after tone, there is a shock. So we tested this attached fairing conditioning in non-enriched and the enriched case. Those were exposed to the lead. So this work was published by our lab. So in this one, the female, we did the experiment in the female first, because we know that there is a difference with behavior in the males and females. So first, to do this experiment in the microbiome, I choose to select the females only. So in this paper, at day 2 and day 10, so in the EPN non-enriched, there is a memory deficit, day 2 and day 10, in compared to the enriched rats. So based on that, I designed the experiment. What if we change the microbiota from the enriched pups, enriched animals to the non-enriched? So for that, we designed the experiment. Here, we took the enriched donors, 6 in a cage, all the animals, and we choose non-enriched 9, 3 cases, 1 case with 3 animals. So total 9, and we took 6 behavior control of the non-enriched. So total 21 animals, and I designed to do the oral gavage, collected the fecal pellet from the enriched caged rats, and make the solution and the oral gavage to non-enriched animals for 4 times a day, 4 times in a week for 4 weeks. So during the experiment, I collected the fecal material at the time of the weaning. At that time, all the pups were the same with the mother. And then after 55 days, putting the pups non-enriched in the enriched environment, we collected the fecal material at that time to see the difference of the microbiome of the enriched and non-enriched environment. And then after the FMD, fecal microbiota biotransplantation, we collected the fecal pellet from the non-enriched 9 animals and the 6 animals with the enriched to see if there is any change in the microbiome. And at the end of the study, we collected the fecal pellet again at the 4th time to see the stability of if there is any change, the microbiome is stable or not, because that is important. So we first measure for the memory testing if the microbiome changing that improve the memory in the non-enriched animals. Then I plan to do the 16S. I did the 16S ribosomal sequencing to see the microbiome population and did the short chain fatty acid analysis. If the microbiome change, the second metabolites of the bacteria, whether they change or not. So it is the memory test. This is on the x-axis is the percentage of the freezing. So in the behavior controlled non-enriched animals, there is at day 10, because day 10 is the most important to memorize the shock and the tone and shock. So at day 10, the non-enriched element, they lose their memory. They didn't memorize. There is a shock after the tone. And in the donors or enriched animals, they memorize everything perfectly. And the recipients, they were non-enriched, they improved their memory equal to the enriched animal. So that is very exciting to see the results because the population, we took the 6 animals to collect the fecal pellet and the oral gavage to the 9 animals. So this started as a recipient of the 9 animals and the donors 6 animals. So that was very exciting result to the further to check the microbiome population. So here did the 16S ribosomal sequencing. So in the weaning, the microbiome diversity is very low. And after we putting the enriched or the non-enriched in the environment, so in the enriched environment, before the FMT, the microbiome diversity increased from the weaning in both pre-FMT and the after FMT in enriched environment was the same. And if you see in the non-enriched pre-FMT, the diversity was low, and after FMT, it increased further. So it means that the transplantation FMT did work, and the microbiome indeed changed in the non-enriched animals. So here I further studied which phylum or the microbiome of the family actually changed in the non-enriched animals. So here I found the phylum Firmicutes, that is anaerobic bacteria, and they are very much abundant in the gut microbiome, and they have very important role to produce the short-chain fatty acids. Mainly fatty acids are butyrate, propionate, and So, this phylum, for the Firmicutes, that was very bacteria population very low in the beginning, and it increased the enriched, and the further increased the enriched, and the low was there was low population in the non-enriched, and it increased in the after FMD. So further, I go in the class and the family and up to genus level, and the same results were there. It increased in the after FMD in the non-enriched animal. And then I looked for the what they generate. They generate the butyric acid that's enhanced the learning and memory, and this butyrate is the has shown as deacetylase inhibitor, and also anti-inflammatory, has the anti-inflammatory effect. And I looked for the further to compare what are the microbiome in the pre-FMD and the post-FMD. So there was increased in the bacterial population after the FMD in non-enriched animals. So here is the I did analysis of the short-chain fatty acid production of the 9 short-chain fatty acids, and I found in the butyric acid that was increasing in the pre-FMD and the post-FMD in the non-enriched animals. So in further experiment, I designed to repeat the FMD experiment to take more animals and to show to validate this pilot experiment. The results we already saw that the behavior is changed, but on the levels of the microbiome and the secondary short-chain fatty acid, we need to show with the more animals, the butyric acid is indeed changed in the after the FMD in the non-enriched environment. And then I plan to do the RNA-seq with the different organs of the different tissues of the brain and the metabolome study. So and this work is done by in the Dr. Snyder's lab, and they have very established model of the lead exposure of the animals, and I'm very thankful for the PRCCEH for funding me for the work, and Jefferson and the UPenn. Thank you.